Hello, my name is Paula Andrea Muñoz Rodriguez. I am a senior student at the University of Puerto Rico, Rio Piedras campus, and today I would like to thank you for taking the time to review my poster titled The Effects of Oxytocin Intranasal Administration in Exercise and Anxiety-like Behaviors in Rats. First of all, I will start by saying that oxytocin is a neuropeptide primarily synthesized in the hypothalamus and is commonly associated with social behaviors, stress responses, and drug addiction. The anxiolytic effects of intranasal oxytocin and aerobic exercise had been studied separately. Nevertheless, the effects of both treatments together and the molecular interactions mediating these effects remain unknown. For this reason, we wanted to examine whether oxytocin intranasal pretreatment potentiates the anxiolytic properties of exercise in adult male Sprague-Dawley rats. And this first hypothesis is based on several studies that supports that intranasal oxytocin administration and voluntary wheel running are treatments endowed with anxiolytic properties. This hypothesis is also based on studies from our laboratory. We observed that there is an interaction between the endocannabinoid system and the oxytocinergic system, modulating anxiety-like behaviors. In addition to the studies that suggest that the endocannabinoid system modulates anxiety-like behaviors, there are other studies that suggest that the presence of the CB1 receptors in the mesolimbic regions is crucial for voluntary running behavior. Additionally, studies had shown a colocalization between the CB1 receptors and the oxytocin receptors in these mesolimbic areas. And this is why we wanted to understand if intranasal administration of oxytocin will produce a higher expression of CB1 receptors in these areas, causing an increase in exercise performance. For these reasons, and because the presence of oxytocin and endocannabinoids in the limbic, mesolimbic, and endocrine systems has been associated with the anxiolytic response, the purposes of the present study were to examine whether oxytocin intranasal pretreatment enhances voluntary wheel running behavior and potentiates the anxiolytic properties of exercise in male Sprague-Dawley rats. Additionally, we wanted to characterize the oxytocin receptors and the CB1 receptors expression within the limbic, mesolimbic, and endocrine regions as consequence of the running behavior. Now I am going to show you a diagram of the experimental design. Before carrying out the experiment, the animals went through a period of quarantine, a period of handling so they get used to human touch, and a 9-day period of habituation, so the rats can get used to the environment and the experimental conditions, with the exception of that they were not given oxytocin. We only administrate them intranasal infusions of the vehicle that in this case was saline. I want to stress out that the habituation is done to reduce any type of stress that a novel environment may cause to the rats. Once the habituation period was over, we start the experiment. The experiment period lasted 10 days, and to carry out the experiment, rats were divided into two groups. The first group received intranasal infusions of oxytocin in our vehicle. The concentration of the oxytocin was 1 microgram per microliter, and the rats received a dose of 20 microliters. To be more specific, we provide them 10 microliters per nostril. And we gave them the dose before the group was exposed to the running wheel for a period of 2 hours for 10 consecutive days. The second group received the same dose of oxytocin or vehicle and was not exposed to the running wheel. On the 10th and last experimental day, after the animals finished the cage exposure session with or without the wheel during the 2 hours, the animals were submitted to a 60 minutes restraint period in a plastic cylindrical restraining tube. Once completed, we use the light under box as a behavioral paradigm to measure the anxiety-like response. Finally, to conclude the experimental design, the animals were decapitated in order to do some Western blots. Now, I am going to show you the results of the experiment, and in this first graph is represented the performance of the rats in the wheel running behavior. In the y-axis, we have the total amount of revolutions made by each rat in a total period of 10 days. And in the x-axis, we have the representation of the groups. The control group received the vehicle infusions, and the experimental group received the intranasal oxytocin infusions. As we can observe, the results show that rats treated with oxytocin exercise more than those treated with the vehicle. After doing a t-test, we obtain a p-value equal to 0.01. This second graph represents the anxiolytic response to chronic treatment with vehicle or oxytocin intranasal infusions. The results indicate that the animals that receive both treatments, oxytocin and exercise, spend less time in the dark side of the light under box, suggesting that oxytocin potentiates the anxiolytic effects of exercise. However, we do not avoid the fact that there is a high variability in behavior. Finally, here is the representation of the results of the Western blots. These were made in the pituitary gland, and as we can see, in the control and experimental groups are represented in the x-axis, while the y-axis represents the amount of proteins of the oxytocin receptors. These biochemical assays of the pituitary gland from both controls and experimental animals show that oxytocin treatment significantly increased oxytocin receptors within the pituitary gland of rats exposed to the running wheel. In contrast, the animals of the sedentary groups that receive oxytocin and the animals that exercise and receive vehicle showed a lower expression of oxytocin receptors. A possible explanation of these findings could be that exercise upregulates oxytocin receptors expression in specific regions related to oxytocin regulation when both treatments are provided. Nevertheless, we are currently examining oxytocin receptors and CB1 receptors expression in other brain regions associated with anxiety-like behaviors to better understand the neurocircuitry underlying this anxiolytic response. Through this presentation, we have seen that there's a trend that oxytocin enhanced voluntary wheel running behavior. Biochemical assays showed that animals treated with oxytocin and exercise had a higher expression of oxytocin receptors in the pituitary gland of rats when compared to the sedentary animals treated with oxytocin. And we also have seen a trend that oxytocin could potentiate the anxiolytic effect of exercise. Future biochemical studies are required to examine the potential crosstalk between the oxytocinergic and the endocannabinoid systems as mediators of exercise performance and all the implications these can have as treatments for drug addiction, sedentary lifestyle, and anxiety-like behaviors. Finally, but not least, I would like to thank you for your time. I acknowledge the National Institute of Health and the Neuroscience Research Opportunity to Increase Diversity program. I will also like to thank my mentor and principal investigator, Dr. Carmel Maldonado Blar, and my lab mates that contribute to this project.